Thank you very much, everyone. It's a very tough gig to start this presentation after such a nice talk, particularly showing a lot of the issues that are in the industry. So I hope that with this talk, you'll see all of the efforts that we're doing at Bio-Techne, and I will be very keen to hear your feedback as well. So my name is Vanessa, and I work in our Oxfordshire office. Today, I will speak to you about what we have been doing for the field of spatial biology in terms of antibody validation. So, as you know, our company has been producing antibodies for many, many years. We have been in the industry for more than 45 years. And the first antibodies that we released were in 1989. So it's a long journey that we've had, and a lot of knowledge accumulated in the field. We have a large portfolio of proven products. So we produce proteins. We produce, in particular, in part of the proteins, antibodies, loads of small molecules, immunoassays, diagnostic tools. One of the areas that I focus on is also automation. So automated Western blots, automated ELISAs that will be ready within 30, 60, 90 minutes, depending on the technology, automated charge analysis, and also size analysis, sorting of cells, etc. But today, I will speak to you about more of our COMET, which is one of our automated instruments to be able to do high throughput screening for spatial biology. And so what we're looking here is our efforts to facilitate the field of spatial biology. So think about it in this way, through your upper respiratory tract, you inhale some pathogens. To your palliative tonsils, you will, make contact with that pathogen. There you will have, particularly in this image, which I'll explain in a second, in the germinal center, you will be able to have B cells that will start producing antibodies. You will have T cells that will be able to fight the infection. And yes, you could use maybe Western blots. You could use ELISAs to get an idea of what is being expressed and at what concentration. But to be able to really understand the phenomenology of what's happening in that tissue, you really need to have spatial information. So what you're seeing in this picture is actually that germinal center. And with CD20, we are detecting our B cells. With CD3, we are detecting the T cells. And with CK, cytokeratin, we're detecting the particular epithelial cells that you can find in the tonsils. And here, actually, we don't only have 3 biomarkers, which would not be that interesting. We actually have a 10-plex panel. It's just that to not overwhelm the eye, we just presented only 3 channels here. And all of these data, what we're doing is published. So what you're seeing here has been peer-reviewed as well. So what you can see is we can detect the particular locations of those cells, and that will be able to inform us on the circumstances around the disease or the disease state. Now, in spatial biology, you'll see a lot of images when people are first starting where everything looks oversaturated. So people may have tried to do, 3-plex, which is what I was showing those 3 channels before, and everything looks oversaturated. And so to be able to do successful spatial biology, you really need to optimize your antibodies. Getting good antibodies is crucial, but also understanding, the assay optimization, getting the right dilutions as well. And so I'll show you the technology that we're using to fast track this process to be able to measure 40 biomarkers all at once. And so here you have the COMET, which is using microfluidics. You can utilize unconjugated primary antibodies. You can do sequential immunofluorescence, which means that you're going to stain, you're going to image and elute, and everything will happen automatically, hands-free. You will have a rapid binding time, so it's crucial to have very good antibodies because the incubation will be 4 minutes in that flow cell, and the elution will happen really quickly, so you have to have fast dissociation as well. You can add 4 slides, and in each of these slides, you can measure a 40-plex. And so what we're doing here at Bio-Techne is we do first, antibody selection. So we'll start with IHC tested antibodies from our companies. We'll do a specificity validation. So we'll try a positive and a negative tissue sample. We will do subcellular localization, and we'll check the overall expression pattern. Now, to verify the sensitivity, we will try for heterogeneous tissues. We will check the dynamic range across the cells and the tissues. And to test for the biological robustness, we will confirm at least 2 additional sample types. And so what you can see here at the bottom is checking for kidney, liver, and tonsil as well. And this, the biomarker is collagen 4 alpha 1. And so let's have a look at our process, at our workflow. So first, we will immunize the animal. We will obtain the hybridoma, or perhaps just using the B cells. And then we'll do a high throughput subscreen in our microtiter plates. We will express, we'll purify, we'll characterize, and only then will we have our new antibody. Now, for the subscreen, we try ELISA, ICC, IHC, and Western blot, and then, only then, do we also try the COMET. So we utilize multiple applications before we fast track it to the COMET. And that is very important to ensure that the antibodies are very robust. For the characterization, we try multiple tissues, multiple assay conditions, and we check for signal background as well for the ratio. And so let's have a look here at aquaporin 4, and let's look at some real data. So once we are trying for our leads in our screen, we will be able to see, we can start with, say, 48 leads. And then as we pass it through, say, ICC, IHC, and then all the way to the COMET, we will end up only with 4 leads. From there, we will then select what will be our new antibody. So we really make sure to remove as many of the antibodies that will not be, perfect to be going to the market. Now, this will be very, very marker dependent, very antibody dependent. Sometimes we will try different for different markers, and we'll start sadly with only 4 leads, and then from those 4 leads we'll end up with just 1. And so, after all of this process, sometimes we're very lucky to be able to say we just got a very good one. So here I'm showing for CD14, for macrophages. We tried initially for the screen on ELISA B, we tested 11 screen, and then here at the COMET, we only had 1 that was good enough. And so again, I want to show you a little bit about that one that we decided that was good enough. We then decided to characterize it further, which is what we normally do. And what you can see here is that after, we try different concentrations and we check first the binding, then after the elution. And if you see that if it is still bound after the elution, which is what you can see at the bottom, then we have to decide that, we need to tweak the concentration, or we might have to say that for certain organs, we will not be able to validate it for our COMET assay. And so we also, when we don't have to start from scratch, we also take some of our antibodies that we have already validated, that we have in our catalog for IHC, and we test them there for the COMET. So here you can see many examples. Here in the column, you will be able to see that we had quite a high concentration, so 15 micrograms per milliliter. If I go back just a second, you can see that for the colon, what we started was simply our IHC concentration and then diluting it down. So you can see we end up with a dilution series and decided that 0.25 was just the right spot when it's not oversaturated. But then sometimes for other markers, you will see that you start with 15 micrograms per milliliter, and that's actually just right for the comet. It's not oversaturating. And so going down is not always the case, or just starting with low concentration is not always the case. You want to start with the concentration that you use for IHC and start diluting down. And so in this case, I'm going to mention something on some data that we had for CD34. In this case, we are looking for the expression in these cells, and as you can see, we are detecting it as a kind of vascular marker, and in this case, you can see it very clearly also for angiogenesis in tumors. And so we have a variety of examples to these ends, showing different antibodies and how we're able to certify them and validate them for the various applications. So putting it all together, this is a slide, which is why I didn't start with it at the beginning, because our eyes get completely overwhelmed. And here we only have a handful of biomarkers. But if you can imagine, sometimes when we try to show our 40-plex just in one image, it's too much. But yeah, here you can see an example with a handful of markers, how we can put it all together and ensure that you have as much information as you need with the spatial context as well. So thank you very much, and happy to take any questions.